Is it recording now? I think it is.
So, yeah.
There we go. Okay. Yes. I know which platform I'll be using next time.
Yeah, Sayali sold you.
Yes. Awesome. We'll wait like 1 more minute. It's 6:05 and then we can get started.
Sounds good.
Okay, awesome. We can go ahead and get started. I think like a few more people might join in as we keep going. But yeah, but hey guys, well thank you all for so much for coming out to our first Today in Scientific Discovery session. I just wanna say that I'm really happy to see that you all are so excited to kind of learn about the research going on. UTD has been recognized as one of the like leading research facilities and institutions in the nation, and we at Molding Doctors and I personally wanted to highlight all the research endeavors that are taking place right around our own buildings and right around our own classes. So today, it's really a pleasure to invite and have Sayali and Serena here from the Biomedical, Microdevices, and Nanotechnology Lab at UTD to present their research with us. They put together an excellent presentation, and I'm really excited for them to share this with you all. So I did want to mention a couple of things before we get started. So for all of you who attend, I will make sure I keep an account, and you all will get 3 MD points for being here. In addition to that, I also wanted to emphasize that this is not as much an interest session as it is an educational opportunity for you all to learn more about the research that is happening around UTD. So just keep that in mind. And yeah, so I'll go ahead and give it off to Sayali and Serena. Y'all can go ahead, introduce yourselves.
Hi guys, so my name is Sayali and I work at the Biomedical, Microdevices, and Nanotechnology Lab at UT Dallas. I'm a 4th year PhD student and that's my PhD candidate in bioengineering. And today I'm going to be talking about sweat-based biosensing, which is the basic overarching theme of our lab, of the research that goes on in our lab. So a little about me, I got my bachelor's in biotech, Bachelor of Engineering in Biotech from Mumbai University in the year 2016. And the following fall, I came to UT Dallas and I started my master's. I did a master's with thesis where my goal was to develop sweat-based technologies for disease diagnostics. And probably a year into my master's, I started my PhD, and hopefully I'm on track for finishing up or graduating by summer 2021. So all my research work in my PhD and my master's has been conducted at the Biomedical, Microdevices, and Nanotechnology Lab, or BMNL for short. And my advisor is Dr. Shalini Prasad. Now, a lot of you might have heard her name. So she's a professor and the department head of the bioengineering department at UT Dallas. And our lab is located in this very beautiful building, which is located on campus, and it's called the Bioengineering Sciences Building. So in this presentation, I will be giving you a little background about what sweat-based biosensing is and why is it so relevant. I'll be giving you some examples about literature that exists in this field for sweat-based biosensing, and also some sneak peek about the research from my from my lab, which is the BMNL, and specifically would be what is in my dissertation. And finally, I will talk a little about the challenges that still need to be tackled for this field, in this field, and then I will conclude with some future work prospects. All right, so one of the most recent and important breakthroughs in biosensor technology is wearable biosensor. Now, this wearable biosensor has attracted considerable attention because of its potential to change the traditional healthcare tools and the hospital-based care system. There is more need for transitioning from these hospital-based or clinic-based systems to a more home-based or personal medicine approach. And this will also reduce healthcare costs and also save up on valuable diagnosis time and help with early detection. And all of this is possible by incorporating miniaturized wearable and implantable biosensors. However, when we think about the progress of shifting from these gold standard technologies that we have to a more personalized approach, there needs to be significant research progress in new materials, in interfaces, circuit design, data processing, and business models. So point-of-care technologies are very central to the idea of doing this shift, going from healthcare, traditional healthcare tools to personalized medicine. So to define point-of-care, these are tests that are performed in close proximity of clinics and hospitals, which basically assist the healthcare professionals with diagnosis. Some of the emerging areas where these POC technologies are booming are detection of sepsis, detection of stroke, thyroid testing, DNA testing, lifestyle monitoring, and endocrine testing. So a little economics about how the global POC market is trending. So it's estimated to rise to $67 billion by the year 2026. So a lot of factors are attributed to this global increase in the market share. And it's because there's still a high prevalence of infectious diseases in developing countries. With the lifestyle that we lead, there's an increasing incidence of lifestyle disorders like diabetes, hypercholesterol, etc. There's also rising preference for home healthcare across the globe. And the one that interests us the most as scientists and researchers is growing advancements in this POC technology area. So biosensor is key to these POC technologies. So what exactly is a biosensor? So you can call biosensor as basically a technology which has a biological component and a physical chemical component. So the biological component can either be an enzyme, an antibody, a nucleic acid, or a cell. So these are usually specific to the target biomarker of our interest. For example, if you have a glucose monitoring system, your target biomarker would be glucose. And then you would use either an enzyme, or actually for glucose, you use an enzyme, which is highly specific for detection and binding of the glucose. And whenever there's binding, the physical chemical aspect of this biosensor converts it into a readable output so that you could get a signal that corresponds to the amount of concentration present either in blood or serum, or in our case, sweat. So a little history about how the field of biosensing and wearable biosensor came into place. So the first concept was introduced by, can you see my pointer? Okay, yeah. So the first concept of enzyme electrodes was suggested by Clark and Lyons in 1962, and it was a basic glucose biosensor where they had glucose oxidase as the enzyme. And then following that, basically you can see over the period of 1963, 73, 80, there were a lot of glucose analyzers. With the advent of immunosensing, they realized that you could do so much more when you incorporate immunocomplexes or antibodies for detection of specific biomarkers. Later, in 1987, they were moving from being bulky electrodes to more user-friendly or small rapid devices. As you can see, the glucose test strips were developed in 1987. GlucoWatch is one of the first FDA-approved wearable biosensor, and it was used to calculate glucose from sweat. And then following with the invention of nanotechnology in the early 2000s, you can see this revolutionized this field of wearable biosensing. By using gold nanoparticles and other nanocomplexes, you could create sensors which were 10 times more sensitive, and also you could incorporate it in different aspects in order to create a more user-friendly approach for wearable system. So as you can see, there are some examples so this was all the advancement in the last decade, basically. So you can see there's like tooth biosensor, there are contact lens biosensors, colorimetric biosensors, sweat microfluidic biosensors, tattoo-based biosensors, and mouthguard biosensors. So you can see how different all these sensors are. And if you look into, if anyone's interested, the source is right here. And the author talks about how advanced this technology has become and how they are able to be very specific for a target biomarker, and also how big of a contribution this was to the field of diagnostics. So later, so this is a slide which talks about the background in the current technology state of the art. So as you can see, there has been rapid development. And what I would like to point out here before I jump into sweat-based biosensing is that the one that intrigues me the most are these tear-based biosensors, or as you can see, smart contact lenses, which they developed for detection of glucose. Now, it's a very interesting approach of using a contact lens as a sensing vehicle and incorporating in either as contact lenses or as eyeglasses, as you can see here. And this research right here is from our own lab, and it's about wearable diagnostics for detection of cortisol and interleukin-6, which is a cytokine inflammatory biomarker. As you can see here, also there's some example of self-powered textile-based biosensors, which is for detection of lactate, and it's a sock-based sensing system. So this is just an overview of what the field has come up to, and there's still a lot to explore, and there's still a lot of research going on in this particular biomedical field. So why sweat-based sensing? So the whole idea is that sweat-based sensing offers a lot of advantages with respect to performance, comfort, functionality, and being economical. So for the performance, it's sensitive, it can be made reusable, and it's rapid. For comfort, it's non-invasive, as you don't have to prick the patient to get a sample. It's pain-free, and you can work with low sample volumes. And usually the sample collection is passive, so the user does not have to take extra efforts to go and collect a sample. They are very advantageous according to their functionality, because you can perform combinatorial detection of different analytes. For example, you can do glucose, lactate, cortisol all at once instead of removing blood and then testing it 4 different times. You can also do label-free detection. That is, you don't need extra fluorescent probes in order to, or extra processing in order to go and get a readout about the concentration of the particular biomarker of interest. And also it can be easily coupled to microprocessors, making it truly portable. And also it's economical because it has cheap cost of production, and also it requires low power. So it's easy to miniaturize and build small economical sensing platforms. So there are a lot of targets that you can detect using sweat-based systems. And you can see this is just a basic summary of what sweat contains and what you can detect. So you can either detect electrolytes, and the main application is for identification or diagnosis of cystic fibrosis, and also to monitor hydration in general. You can also use sweat for detection of small molecules or metabolites like glucose, urea, creatinine, uric acid, cortisol, ethanol, etc. So the main applications are either diabetes screening, kidney failure evaluation, wound healing, stress evaluation, and drug dosage control. Some other fields are xenobiotics, which are detection of foreign substances in the body. So they might be either heavy metals or drug residues, and usually it's carried out for product safety evaluation and forensic chemistry. And you can also detect proteins and nucleic acids, so specifically interleukins that we just talked about. And their main application is detection of infection, as we saw with respect to sepsis or inflammation, or in general inflammation, or regards to a chronic disorder. And finally, you can also look at physical fitness, and this is huge in the field of athletics, as we all see. And you can detect ammonia and acetic acid and also lactate to evaluate the athletic performance and also for body odor. So that brings me to some of the examples of the research that's currently going on in the research labs across the whole country. So this is a very interesting panel of epidermal biosensors for real-time monitoring of sweat chemistry. So as you can see in the first example here, it's a fully integrated wristband-like device. So by fully integrated, what I mean is that it calculates the concentration and it processes the signal output and gives you a readout, which does not need any extra processing. So it's basically like an all-in-one sensing system. And that sensor array can detect glucose, lactate, sodium, potassium, and temperature during exercise. The second example is a basic colorimetric sensor, where, as you can see, it detects glucose, pH, and humidity. And it's this adhesive tattoo-like patch that you can just put onto your skin surface. The third layer here is, it's basically like a Band-Aid sort of design, where it's a waterproof band and it has a porous system which uptakes the sweat. And basically what it does is it's used for monitoring of glucose, and it's applied to the forearm for sensing during exercise so that there's enough sweat volume collected, and you can basically evaluate the concentration based on the color change. Now this little research bubble here is from our very own lab, and it's basically for detection of cortisol and interleukin-6, as we saw in the first figure. And this is one of the pioneers of our lab, where we use something which is known as room temperature ionic liquids to increase the sensitivity of the biorecognition probes. So biorecognition probes are basically the biological elements of the biosensors that we talked about. So with incorporating RTILs, we increase the life of the sensor, and the sensor can perform detection over the period of 24 and 48 hours. So that's great with respect to self-monitoring, where you need to continuously evaluate the different sweat biomarkers for longer periods of time. Some other examples include, for example, H, where they have glucose and uric acid, and also lactate and urea. And as you can see, we talked about ability to detect multiple analytes in one go from just a small sample volume. And this can be really observed in all of these research, where you can see that they're doing multiple analytes, and it offers a very comprehensive evaluation of the sweat chemistry. And finally, this is this is used for detection of tyrosine, and it's actually based for early detection of melanoma, as by using tyrosine as the biomarker. And it's, as you can see, it's very similar to a Band-Aid, and it's a simple Band-Aid-like patch. So that brings me to the research from the BMNL lab, and specifically my dissertation topic, which is SLOC, that stands for Sensor for Circadian Clock. So basically, this is a sweat-based chronobiology tracker. So circadian rhythm is basically the innate sleep-wake cycle present in your body, and it controls when your hormones are going to be secreted, when it's the time to eat, when do you go to bed, etc. So as we all know, especially the audience of this this seminar, that we lead a very hectic lifestyle. So according to the CDC, 50 to 70 million adults are affected by chronic sleep wakefulness. What I mean by chronic sleep wakefulness is by not having a fixed sleep-wake cycle. And the reason why we're interested about the sleep-wake cycle is because having an absent or dysregulated circadian rhythm has a lot of implications. It can increase your risk for diabetes. It can hamper your cardiac functioning. It can have a lot of metabolic and neurological implications. So the idea is to use a sweat-based system so that you can go and measure these sweat-based biomarkers and track the user's sleep-wake cycle. So this is the recently published work from Lab on a Chip, and it came out just about a month ago. And this basically shows an overview of what my sensor looks like. So what we're trying to do is we're trying to detect these biomarkers, which are cortisol and DHEA. Now, cortisol and DHEA are basically adrenal corticosteroids, which are secreted by the adrenal cortex. And they have a very peculiar rhythm of secretion, and that exactly maps your sleep-wake cycle. So by monitoring the secretion of and the levels of cortisol and DHEA, what we're trying to do is we're mapping the user's chronobiology, and we're trying to see if he suffers from sleep wakefulness or if there's a circadian dysregulation, which usually happens when you either work night shifts or you travel across different time zones, or in general, if you have circadian disorders. So the reason why they are two different patches is because cortisol is present in eccrine sweat. So eccrine sweat is something that basically produces sweat as a response for thermal regulation, which is maintaining your body temperature. And it's present all over your body. So your palms, your soles, your back, etc. And DHEA is more of a secondary sexual characteristic, and that's why it's secreted in your axillary area. And that's why we have these two different patches, basically, for collection of sweat and detection of these relevant biomarkers. And what's unique or what's novel is that we have a unique serpentine electrogeometry, which gives it additional flexibility so that it can conform to the user's movement, especially when the user is sleeping, so that it does not distort the signal, and it also is able to robustly detect these biomarkers. So that brings me to the challenges that still needs to be tackled before this field can become a gold standard for diagnostics. So some of the challenges that are associated with sweat-based sensing are device on-body use, where stability under uncontrolled conditions like excessive sweating, excessive temperatures, rather very cold temperatures, or not enough sweating also. So we need to make sure that our sensing system is robustly operational, and also that it doesn't need frequent calibration, because you wouldn't want to sit there and like recalibrate your machine again and again to incorporate your sweating rate at that point of time. And also we need to make sure that there's proper fluid sampling system so that appropriately the sample is collected and analysis is done real time. The second and the most important is system integration and hardware. So in order for it to become a truly wearable, we need to think about how we're going to incorporate it with the suitable hardware. We need to make sure that it consumes low power, it's safe to use on the skin, and also usually when you design these systems, there's a trade-off between how much energy is consumed and how much data is collected. And you need to make sure that you're collecting enough data so that you can give the reader and give the patient the output that, hey, this is your cortisol level or this is your DHEA level, while also maintaining low energy. And also another issue that we're trying to solve in our own lab is how can we do direct on-body data analysis so that when we go and read and the output is actually what it means, and there's no factor that we need to subtract, or there's nothing extra that we need to incorporate in order to see a clear value of the biomarker. And also making sure that because sweat contains a lot of interfering species. So there might be excessive amounts of lactate if someone just exercised. So we need to make sure that these other molecules that are present in sweat, they don't go and interfere with your molecule for detection and give you false positive or inaccurate readouts. And also that brings me to accuracy and stability. We need to make sure that our sensing system is stable across the period of time that we are using it for. And also it's anti-fouling, because your skin, it contains a lot of cell debris, hair, and like skin and like bacteria also. So we need to make sure that the sensing system that's in contact with our skin does not get rusted, does not get soiled, or like it stops giving you the readout because it's in contact with the skin. And also we need to make sure that the conditions that we're putting the sensor at, they're stable enough for the biological probe to work and perform the detection. Oh, and also one last point, that we need to make sure that they are correlated with the blood biomarker levels, because usually blood or serum are the gold standards that clinicians use. So we need to make sure that whatever biomarker we're trying to detect, they have good correlation with the actual blood slash serum biomarker levels. So to conclude, wearable sensors are promising solutions for the field of wearable diagnostics, and you can use it for monitoring metabolites, electrolytes, heavy metals, drugs, and hormones. But it will require extensive and successful validation with human testing. And also we need to improve our current understanding of the clinical relevancy of the sensor information that we receive from these sweat-based technologies. And also a little trend is that now these sweat-based sensing technologies are getting more streamlined. So they're moving away from being just a normal wristband or like a dermal patch to more incorporated into textiles and like fashion accessories that can blend into the wearer's daily life. So that brings me to the end of my presentation, and I think I'll hand it to Serena for the rest of the presentation, and then we can take questions in the end.
Yeah, should I share my screen, or do you want to control it on yours?
I can control it. Yeah,
okay, I'll just let you know.
Just let me know when, like, just say next, and I'll like switch.
Okay. Okay, hi guys. So my name is Serena Bhadsavle. I'm a senior this year, majoring in biology. And I've been a part of BMNL for the past 2.5, almost 3 years now. So today I'm going to be talking a little bit about my experience in BMNL and the contribution of myself and other undergrads in our lab. Go ahead. Okay, so one of the biggest contributions that undergrads have in our lab is helping out with experiments. So in particular, one of the experiments that we do is called a calibrated dose response, or CDR. And basically what we're doing here is we're feeding our sensor increasing concentrations of whatever molecule that we're interested in. So in this example right here, in this picture, the bar graphs, the red, the blue, and the green are all have increasing concentrations of cortisol. And you can see that as we increase that concentration, we get an increased output from our sensor. And so this experiment is really critical, not just understanding the protocol, but also understanding the implications of what you're doing. So go ahead. Okay, so one of the other roles that I have as an undergrad in the lab is preparing sensors. So before we can run a CDR, a lot of times we have to prepare our sensor in some way so that it makes taking measurements easier. So in this pictorial, this is kind of what our sensor looks like, kind of an enlarged graphic of what our sensor looks like. So this gray bar right here at the bottom kind of represents the sensor material. So we have different types of materials that we use with sensors. So this can be variable. The gold, the yellowish gold bar right here represents our electrodes. And then the molecule in the white boxes right here is our linker, which basically connects our antibody, which is in blue, with our gold electrodes. And the antibody, like Sayali mentioned earlier, is what binds our molecule of interest. So in the previous example, we were talking about cortisol. So we would have an anti-cortisol antibody binding to our cortisol. And the interactions that are happening in this kind of sandwich of binding, if you will, are what allows us to collect measurements for our sensor output. And so incubating both of these elements, the linker as well as the antibody, sometimes takes a couple of hours. And so that's a pretty big contribution that undergrads have, is just preparing those sensors to get those measurements in our CDR. Other ways that we kind of prepare our sensors are just extending some of the wires on our sensors just to make it easier. So we haven't actually showed a picture of what one of our sensors actually looks like, but they're pretty small. They're like pretty tiny. And so sometimes adding those wires to make it easier to collect data helps us out quite a bit. So moving on to the next one. One of the third roles that I have in the lab is just learning about different tools and technology that we use in the research process. So I think as an undergrad, it's important to kind of involve yourself in as much of the research experience as you possibly can. And so you're learning things like wet lab techniques, and you're also learning about different types of software that we use to collect data, and even create things like this box that I have right here. So this box is something that I've actually created using a program called SolidWorks, which some of the undergrads here might be familiar with. But SolidWorks is basically a CAD program that allows you to design and 3D print different objects. And so what I've created right here is kind of a prototype for a sensor platform. And you can see our sensor kind of peeking through that hole right there. And so learning about just these different types of technology and different techniques kind of allows you to familiarize yourself with how research works, how the process of testing, experiments, all of that works. So that's kind of a big aspect, kind of a general thing that we learn as undergrads in the lab. So fourthly, and probably the most important kind of takeaway as an undergrad, is just familiarizing yourself with scientific literature. When I first started out in BMNL, it was, I'm not gonna lie, it was pretty challenging to kind of read through a lot of these scientific papers. I didn't have a lot of experience reading them, and it took a while to kind of get through just one paper. But the more that you expose yourself to scientific literature, the easier that it gets. And I've experienced this firsthand. And so I think, just being able to read some of these papers and try to understand them with a mentor like Sayali helps a lot, because if you have questions, she's always there to answer those questions and kind of guide you along through the process. As anyone going into healthcare, scientific literature is not going to go away. It's always going to be present, and it's always going to be a part of your career. So I think, getting involved in research allows you to kind of get that exposure early on and get accustomed to reading and even writing scientific literature. As healthcare, as anyone going into healthcare, it's always important to be continuously looking to the future, looking at how the healthcare field is going to change. And I think that being involved in research is kind of the best way to do that, because you're able to experience firsthand some of the ways that might change the way you practice 10, 15 years in the future. So to all the undergrads here, I really encourage you to get involved in research, to really just take it as a learning experience, learning opportunity to kind of see what's out there and see the way that healthcare is going to change. So those are just four of the kind of main roles that undergrads have in the lab, in our lab in particular. But of course, you want to try to get involved in as much of the process as possible and just learn as much as you possibly can. So with that, I think we'll open it up to questions. We also have our contact information here. If you end up thinking of anything after this presentation, you can jot down our emails, and I'm always open to asking any question, answering any questions. So.
Yeah, I would like to add, so if anyone's interested in working in the BMNL lab, you can shoot us an email. And so for now, for the next 2 semesters, we're going to be virtual. But once we get back up and running, you can come in and we will be able to train you. So hopefully if you're in your 3rd year of your undergrad, that would be the best time to start, because you would at least get a year's worth of experience. So just send me an email and I can patch you through whichever grad student has the best skill set that matches yours, and we can get started. Yeah. And then with that, we can take questions.
Wow, that was an awesome presentation. Thank you guys so much. And that was really, really insightful. And just listening to you all made me just kind of imagine and kind of think about all the possibilities out there for the future. So I think I'm really, really looking forward to everything that comes up in the future as we go on to be in the healthcare field and so on. But I think we do have a question in the chat. So Ariella says, do you foresee wearable technology playing a larger role in patient care, such as doctors receiving real-time sample monitoring from high-risk outpatients? Yeah, so I guess that's the first question.
Yes, so basically what sweat-based or wearable technology does is it bypasses your routine blood test. So what happens in a routine blood test is that the surgeon, for example, they take the blood, and then the blood goes through a lab where there's a technique called ELISA, which is enzyme-linked immunosorbent assay. And that is the standard technique that all the diagnostic labs use to give you those blood panel results that they have. So that ELISA procedure, it takes 6 hours. And that's why whenever you go for a blood test, they tell you that, oh, come back after like 24 hours or like 48 hours. So what wearable technology does is it cuts down that time to like 15 minutes. So within 15 minutes, the surgeon, if it's a surgery that the patient needs rapid analysis of the biomarkers. So I think the best example would be, I remember this, that for thyroid surgery, so whenever there's a thyroid cancer potential or a tumor removal surgery, what they do is basically they remove the, not thyroid, sorry, parathyroid. So they remove the PTH, and then they instantly do a readout of what are the parathormone levels in the surrounding tissue, and then they close the patient up. So basically the patient's open and they're waiting on the blood results to get the levels of the biomarker before it's safe for them to close up the patient. So that's something that our lab is also working on. And what they're trying to do is they're using this technology to basically reduce that time for calculating and like giving output within like 15 minutes. So the doctor knows, okay, like I gotta like act quickly, or like, you know. So basically, yeah. So and the answer to your question would be yes, it is aimed at making diagnosis better, and also at making management of chronic diseases better. So for example, someone with long-term chronic disorders, say even diabetes. So it's a self-monitoring system where the patient does not have to constantly go get blood tests or constantly even prick himself with those glucose test strips. Rather, he can just wear a wearable patch and just go about his day, and it will like let him know when the glucose levels like fall below normal. So yeah.
And another thing to just like quickly add to that, like I'm wearing an Apple Watch right now, and so like something like this, even in the future, can monitor your glucose levels, can monitor your levels of various biomarkers. So even something like that, I think we're definitely going in towards that direction.
I have a quick follow-up actually to that question. Is there like I know you were mentioning it earlier, you all were mentioning it earlier, but in terms of sweat-based sensing, is there a certain like level of sweat that is necessary for like the sensors to even like sense these different biomarkers?
Yes, so the sensors that I showed you, like my specific sensor, it works at a volume of 10 microliters. And basically, when you're designing sweat-based sensing technologies, there's two ways that you can go. One way is where you do not do any stimulation, and it's just collecting sweat passively. So if you go via that route, which is also what I'm taking, usually the patient, when he's in like a resting state, or not patient, or the user, when he's in a resting state, they produce so the rate is 5 to 10 nanoliters per gland per minute. So if you do a calculation, it comes up to between 5 and 10 microliters per like hour or so. So that's why I've designed these sensors where you basically collect 10 microliters of volume, and it's collected over the period of an hour to give that hourly output. And for my application, hourly readings are fine. But there's some applications where they use as low as 5 microliters of sensing volume, and the readout is instant. For example, with respect to glucose and lactate biosensors.
Wow, that's amazing. Yeah, that makes sense. That's really, really cool. Ariela had one more question for you all. She says, with the increasing market growth in wearable technology, do you think additional steps should be taken to preserve the integrity, storage, transmission of medical data?
Yes, absolutely. That's one of the one of the whole big ethical issues, and because with wearable technology, like for example, you don't know who's owning that data. Is it the company that made the wearable? Is it the scientists who like made the technology? So I think there's a lot of issue of where and who, and do HIPAA laws also apply to that kind of information? So that's like a very, I would say like, it's like a gray zone where no one knows what's happening. And I think it's according to the company that's selling them. And if they give out disclaimers that, hey, we're not the ones who are looking at your data, or like they give clear disclaimers that, hey, this is the person dealing with your data. I feel like that would be step one of solving this big problem. But in general, I feel like universities, when we're doing these research, I would like to add, we are very, have been trained, and we're very confidential with how we handle the human subject data. And there's this board called Institute Review Board, which is IRB. So we can all do human subject experiments only after we get approval from the IRB, which is a set of experts in this in general field. Maybe law. Some of them are lawyers, and some of them are like they have invested their careers in protection of these human participants. And that's so every university has that kind of board that makes sure that all the research that we're doing is ethical, and the data that we are getting is stored properly, so that, you know, it's not out there. So whenever we do human subject research, we have to de-identify whatever the subject is or who he is, so that the data does not, we cannot correlate the data to which person that was. So that's one of the things that we do at UTD. So, and like in general in academia.
So is there, in addition to, is there like a board that approves like clinical trials? Do devices have to go through clinical trials as well, just like medications do? Or is there something similar to that? Yeah. Okay.
Yes, so it's similar. The clinical trials for medical devices is very similar to the clinical pharma drug trials. And usually it takes a lot of experimentation, especially with human subjects, to really prove its efficacy. So I think I would like to give the example of GlucoWatch. If you saw in my presentation, I can just like quickly go back, because it's a very interesting example, and I just like remembered it. It's basically, it was a glucose monitoring system, and it was from this biotech company called Cygnus. So like right here, if you see the glucose stuff. Okay, anyway. And what's happening is that this glucose monitor, it was FDA approved. And then all of a sudden, because remember how I said about there can be two routes where you can collect sweat passively. So the second route for that is that you can stimulate sweat. So basically what you have is a light, like it's called an ionophoresis system. So what it does, it supplies a small amount of current, and it activates, and it has usually a sweat stimulating agent. So for the GlucoWatch, it was pilocarpine. So that pilocarpine is basically a drug that stimulates your sweat glands to produce sweat. And by combining the pilocarpine with the current, you can stimulate sweat, and you can get more volume of sweat at one point of time. So what they had is basically, they had that sensing system. And for that, like right here, you can see, they used to detect glucose. So all of a sudden, it just stopped working, and it used to heat up a lot. And it used to give burns to the patients. And also, it had a little alarm on there where the glucose levels, if it went below normal, it would just like give a loud alarm, and this was usually used for pediatric patients. So then the alarms used to be so loud that used to disturb the patient, and like the kids would start crying, and also it would be not accurate. So the levels would actually be normal, but it would just give like a loud alarm for no reason at all. So for that reason, this was eventually discontinued, and that was one of the things that, you know, it was pioneering for the field that it got FDA approved. But then, you know, it's like, is it safe enough? Like, was there enough testing done to make it like, make sure that it's like safe and that it works when it's actually on body? And that's why I presented the little challenges section, that no, like we still are ways behind from where this technology could go and become something like, which is a gold standard. So yeah.
Yeah, that's amazing. Yeah, and that's actually crazy that happened like that. But I guess like that's what's necessary for advances. But in addition to that, do you see like sweat-based sensing? So I know it's like, I guess like what you were saying was it has to do with like enzymes and the fact that like glucose is being converted into another compound, and that's what it's detecting. Am I right in saying that?
Yes. So basically, the glucose oxidase, it breaks it into, it's either, I forgot the molecules, but it's basically it converts the glucose into some sort of substrate that is easy to detect. And that's why when you see that conversion happen, that's when it picks up that, okay, this amount of product is formed, that means the substrate is this much. Because it's a simple like stoichiometric like reaction, like first order.
So do you see like, do you think like that enzyme could be placed in like Serena was saying, like, do you think that enzyme could be placed on like an Apple Watch and like could be used in the future just like a normal wearable device? Or would it have to be like medically prescribed per se?
So about the prescription, it would depend, because you have to also make sure that they're covered in the healthcare. Yeah. But one of the examples that I showed right here was this array. And that was something that, so yeah, basically that was gluconic acid is the substrate. So the gluconic acid is the product and glucose is the substrate, and glucose oxidase is the enzyme. So you can see that this is kind of like going towards that wristband like device, but I'm not sure. I think this is from, I'm not sure. So this paper is in Nature Biotech, and it's from, I think it's ASU or Berkeley, I'm not sure. So I might be wrong, but if you go look at this paper, this is basically a panel in that paper. And they talk about the different epidermal biosensors, and this is from one of the labs. And this is a fully integrated like wearable where they're trying to see detect glucose, lactate, and like temperature. So.
Got it. Wow, that's amazing. That's really, really cool. I guess switching gears a little bit, would you guys be able to, like Sayali and Serena, both give your journeys into research and like why you chose this specific field of research over the others, or what made you interested in this?
I could go first. So I have a like a very interesting, because I switched majors also. So when I started as an undergrad, I was pre-med. So I wanted to go to med school and back in India. So basically in India, the way it goes is that we have a 5-year medical undergrad instead of doing the pre-med track and then applying for MD. So we usually do a 5-year medical undergrad, and then we do residency, and then we do our MD. And then I was also interested in doing engineering, because I was like, I'm good at physics and chemistry and I want to be able to use it. So then that's how I got into biotechnology. And with my capstone project, we made these cellulose nanofibers. And with these nanofibers, the potential application would be using these materials for wound healing or for tissue engineering. So that's how I kind of got into the field of nanotech. And I was looking at different universities to come to the US. And Dr. Shalini Prasad's lab was the nanotech lab at that time. And I looked at her research and it looked very interesting. So then I started as a master's student and I started working on my master's thesis. And I think that's when Serena also helped me a lot. I mean, she helped me all throughout my like PhD and my master's. But with my master's thesis, that's the paper that she presented, where we were trying to make these small, like universal biofluid sensors, because the whole idea was that we could do different non-invasive biofluids. So we could do like sweat, we could do saliva, we could also do urine, but main focus was like sweat and saliva. And then going into my master's, I was like, oh, like I want to do something more with this. So that's how I went and talked to my advisor, and then she's like, maybe you can switch to a master's plus PhD program. And that's how I started doing master's and PhD simultaneously. And I got to work on like one project and like gain all of that knowledge and then apply it towards my actual dissertation, like doctoral project. And that's why I'm hoping that, you know, I get to, I feel like I'm coming close, like it's all coming together. So I'm like hoping that from here, I go on to industry and actually contribute to the, you know, actual like wearable sensing technology or even point of care technologies. So especially in the age of this like pandemic, we need to make sure that we have these rapid kits that, you know, they can tell you, okay, you're like COVID positive or not. So that kit is just an example of how point of care technologies is changing the field of diagnostics. So that's my experience.
That's amazing. Yeah, I was actually, that was one of my questions for you, was like, how hard was it to kind of combine like engineering and with biology and like the medical field, but that's amazing. That's really cool that you were able to do that.
Yeah, so
just, oh, sorry, I'm sorry.
No, go ahead.
Just the last point that don't be afraid of like going and exploring options, because for me, like when I was switching from biotech to biomed, I was like, oh my God, like I don't have any engineering background per se. Like the only engineering background I had was genetic engineering. So it's like, it's not gonna like actually help me do like these devices, but it certainly helped with my understanding of how genetics work and how they contribute to chronic disease and like their manifestations. So all I had to do is just do a few like prerequisites. So I had to take up like some basic like electronic classes and like some signals and system classes. So I would say that, you know, you should like not shy away from doing this just because it looks so complicated, because like everywhere they can teach you, they can train you. It's your ability to quickly learn and like reproduce. Like that's what they're more interested in. So,
yeah, definitely. I definitely agree with that. I think a lot of, as an undergrad, you get very like intimidated almost from research because you think like, oh, I'm never going to understand it. I don't have the right skill set, but you end up learning on the job basically, you know. So going back to your question about, you know, my journey, kind of similar to Sayali, I had interests all over the place. So when I first came into UT Dallas, I was a biomedical engineering major because I knew I liked physics, I knew I liked math, I knew that I liked biology and medicine. And so bioengineering kind of seemed like the perfect conglomeration of everything, you know. And during my first year, I think it was like my spring semester, maybe fall, we had a project where we built kind of a novel heart rate monitor in one of our intro bioengineering classes. And that was what kind of like piqued my interest in particular. And so I remember I went to like the research fair that was going on, and I started hearing a little bit about Sayali's lab, or our lab, BMNL. And because of like my interest from that one project, I was very interested in joining BMNL. And once I joined BMNL, I ended up switching my major from biomedical engineering to biology. But I think what keeps me motivated to contribute towards this lab is just the implications of it. I'm pre-optometry, so I'm hoping to go on to optometry school after this. But even this type of research is relevant to an optometrist as well. You know, Sayali was talking about the contact lens sensors and the glasses and everything. And I think even that's relevant to me as an optometrist in the future. So I think the implications of this lab are relevant to not only medicine, but to, of course, biotechnology, any practicing clinician, really. And so I think that was what kind of motivated me to continue working in this lab. And here I am today.
That's awesome, Serena. That's awesome. How was, I guess, like one last question because we are getting close to time. But for Serena, like, how was your experience with scientific literature? And like, I know you're talking about how it was intimidating at first, but in the end, you ended up doing some writing and like you actually got to engage in that. So how was that experience?
So like I was talking about before, when I first started, like I started during the summer, and I remember during that summer having a pretty hard time kind of reading some of the articles from our lab and understanding just some of the words that they were using and the process of research that was described in these articles. But I think like the more, and Sayali did a great job of kind of like giving me like almost like homework in a way, just like giving me papers that maybe she could explain to me if I had any questions. And so that was really helpful just to get some experience and to have a mentor to explain to me exactly what this is talking about if I didn't understand it. And it sounds crazy, but like the more you do it, the more it really gets easier. And I took what class? It was a biochemistry lab course I took last semester. And in that class, I felt like I actually started to have a grasp on it because of my experience in research. You know, being able to read some of the papers from research kind of helped me in that class. And writing research papers also, like writing sections of research papers has also been pretty helpful because doing it yourself kind of helps you understand what other people are saying too. So I had a little bit of experience writing, you know, materials and methods sections, intro sections, stuff like that. And so I think that is going to help me definitely moving forward in the future.
That's awesome. Yeah. I think for me especially, and a lot of undergrads, it's definitely very intimidating just reading, you know, it's like you don't know what's even going on.
Yeah.
You're right.
I'm not gonna like sit here and be like I'm an expert in reading these research papers because I'm definitely not, but it does get easier and I think it's important to at least try, give it your best effort each time you read a research paper.
Yeah, I would like recommend just asking questions. Like don't be like, oh, like this such a stupid question. That's like, no, it's actually not. Like even when you are done with your PhD and like you're, you know, it's still not a stupid question. You're just not in that area. So it's always like, you know, we always like people who like do research like us, we're always open to answering questions because like that's what we want. Like we want more people to be aware of this kind of technology and like we like more questions. So yeah.
Awesome. Well, it is 7. So I don't want to keep y'all too much longer, but guys, if y'all have any questions for Sayali and Serena, you can go ahead and email them. I know I'll go ahead and link the PowerPoint as well in our email. I'll send that to y'all and you can go ahead and email them with any questions that you have.